Welcome to this mini symposium on ALS and Alzheimer disease. Do metals have a role in neuro-degeneration? Geomedicine is about how natural environmental factors on our Earth affect the health of animals and humans. It is a large and exciting field, but the field is also considerably larger because it can be difficult to distinguish natural from man-made anthropogenic environmental hazards and problems that are gradually and now completely dominating our globe. Warm welcome to Professor Wei Zheng from USA. His presentation will follow after the introduction to the topic of the evening by Professor Jan Aaseth. Thereafter, senior researcher Per Selnes will tell more about the Choroid and Alzheimer disease, and Professor Per Roos about the Choroid, ALS and metals. Postdoc Karin Persson will present new medicinal possibilities for Alzheimer before Jan Aaseth will sum up and open for a short discussion and conclusion. Then the floor is yours, Jan Aaseth.
Here we can see a healthy brain and a severely affected Alzheimer disease brain. But it starts with abnormal amounts of amyloid beta, A-beta, retained extracellularly in the brain as plaques, and tau proteins intracellularly as tangles. These are the characteristics of Alzheimer's disease. It might be even more interested with ALS. We will hear more about that disease from an expert from Karolinska in Sweden, Per Roos. And the characteristics are as follows. It is a rare neurodegenerative disorder that results in the progressive loss of both upper and lower motor neurons, these neurons normally control voluntary muscle contractions. Most cases of ALS have no known cause and are known as sporadic ALS. We think that environmental factors are involved, for instance, toxic metals, for instance, lead, but most probably not lead alone. We will hear more about these findings from Dr. Per Roos from Karolinska. A newly described disease is this Skogalt disease, characterized by neuropathy and encephalopathy with slow progression from about 30 to 80 years or more, and the CSF, cerebrospinal fluid, is characterized by very high protein levels. And here we think that the choroid plexus is insufficient in one or another way. Perhaps the aquaporins, ATP dependent, does not operate as normal. We have found high copper levels and also iron levels in cerebrospinal fluid, and think that there is an insufficient export by the so-called ABC transporters, but we are studying these findings more, and we therefore have invited Professor Wei Zheng to tell us more about the choroid plexus today. We will see from Per Selnes findings on MRI on the choroid, I think. Then I will introduce the first lecturer of today, Professor Wei Zheng. He started at Columbia University and is now professor at Purdue University, a highly ranked university in Indiana, in United States. He has been chairman of several international conferences where I have participated, and he has carried out several experimental studies on this choroid plexus, I guess almost 40 publications. Then your floor is yours, Professor Wei Zheng.
Today, I want to address that the career-long research on the choroid plexus, and especially talk about how that is the metals that induce the neuro-degeneration. So the first, I'd like to give you some background information. What is the choroid plexus? And today I'm going to mention the very important concept called the subventricular zone, the SBZ. Remember, subventricular zone. And we will talk about a lot about adult neurogenesis. Then I'm going to spend quite a bit of time. It's almost half of my talk going to be focused on what we call the evil axis. It's not politically the evil axis. It's the evil axis in the brain. That is, we call the choroid epithelium, the ventricule, the interactive lineage. That is the in the brain. Then I'm going to touch a little bit about the lead exposure and ALS. I think I will leave the lead and the ALS story to Roos. Then finally, but I like to spend some time to talk about the copper. My lab has been spent about 15 years. I try to understand how copper move in and out of the brain, especially how that choroid plexus plays roles in the copper transport. Here is a slice. You will see that is the brain. That this is a tissue. You see if you dissect the animal and see the human, and in the brain, you will see this is a choroid plexus. It's a small tissue. That in the human is about 2 to 3 grams, but in the animal is about several milligram. Then under the microscope, you can see this is a choroid plexus tissue. Okay, that the tissue itself, actually the inside of the tissue is a blood running through the choroid plexus tissue. And the choroid epithelial cells that are facing the cerebrospinal fluid, there's a lot of microvillas. That microvillas increase the surface area tremendously. You think the choroid plexus is only several milligrams, but the surface area is about half of the blood-brain barrier. So surface area is not that insignificant. And this video probably will tell you how important it is. Okay, is a very vibrant, the blood coming through the choroid plexus. And this video doesn't show the pulse in the other meeting at King's College in London. Then we saw that in the human, the probe with the camera insert into the brain ventricles, and also they monitor the heart pulse, and the choroid plexus actually moves around the heart pulse. So in other words, it really affects the choroid plexus related to the systemic circulation. That is how important this tissue is. The main function of a choroid plexus obviously is the cerebrospinal fluid. The secret is cerebrospinal fluid. But in the meantime, it also brings a lot of garbage, and especially the beta amyloids and the out of the cerebrospinal fluid. So we have some evidence about this. And the one more thing I like to mention is that toxic metals like lead and manganese, tend to accumulate in the choroid plexus. We don't know why, but this tissue itself is tend to accumulate this toxic metal. And obviously, this is whenever that happens in the blood compartment, the first reaction in the brain could be the choroid plexus because rich blood flow to the choroid plexus. Now, this is the choroid plexus. Next to the choroid plexus is what we call the subventricular zone. Okay, subventricular zones are quite unique because the cells on the wall is not flat. It have a lot of sensor. This sensor can actually sense what is going on can sense what is going on in the cerebrospinal fluid. Anything happen in the choroid plexus, and the liquid it secretes that will affect the first that brain ventricle, subventricular zone. We have a great interest in the SVZ. Later on, I will mention why, because we found that the tissue that accumulates a large amount of the copper, and because of this, we try to understand what is SVZ. Then check for the literature and Google it. We found out SVZ is a place that generates the new neurons. We all understand that with the new neuronal cell died, the neuron will not grow back. But in the brain, there is two niche places. One is in the hippocampus, dente gyrus. The other one is in the subventricular zone that can generate the new neuron and supply to the neuron to the rest of the brain, and including the oligodendrocytes. Okay, so there's the question we have is that if there is a choroid plexus SVZ regulatory axis, because if you look at the anatomy, it's so close, and also there is a median running through this choroid plexus secretes cerebrospinal fluid, then the reach the SVZ. So the question is always wondering that I think everybody can ask this question: is there a regulatory axis? Okay, to prove this concept, and we have several students in the lab, so we talk about so how can we move forward to understand the axis or the evil axis. And the first that we did is the in vitro experiment. It's very simple, straightforward. You can actually dissect the SVZ and culture it. Okay, during the culture process, can we let the choroid plexus next to it, see what happened in that place? So we did the first experiment that we ever did is use the adult SVZ derived neurosphere. Okay, neurosphere model. Here you just cut into the brain, then dissect the SVZ tissue, and a very elegant procedure. Then you can go put in the cells, the SVZ derived cells, and in the cell culture, gradually after about a week, you will gather the neurosphere. Then with that neurosphere, you identify it, characterize it. Then you have a lot of things you can do, okay? In terms of a toxic exposure, in terms of the choroid plexus co-culture, okay? So first thing we do is that we have SVZ in this chamber. Put the underneath of this chamber. Then you put the choroid plexus, freshly collected the choroid plexus tissue, the rich, vital. Then you put those two things together, then you want to understand how that the neurosphere changes. Okay, then we use the different kind of the markers, and the neuronal cells they can actually derive from the neural stem cells, gradually become neuroblast, eventually become the neuron. Each step you can find out the different kind of markers. Okay, it's a very specific marker. Then you can label it. That's how you see that this is a normal neurosphere. Once this neurosphere co-culture with choroid plexus tissue on the top, you will find this neurosphere grow bigger. And also the cell become the very, very rich, abundant, have the different kind of the cells. Each stage of the cells, you can see this. And the next stage is that you found out, okay, the choroid plexus indeed in the cell culture, they has some effect with the neurosphere. Can we inject the choroid plexus cell, culture the choroid plexus cell into the brain. Then you dissect the subventricular zone, see how that the neuronal cell changed in that area. So that's what we did. Actually, you gave the injection of this needle supposed to be in the lateral ventricle. Okay, the needle just move a little bit here. But you will see this is a setting. We have the animal and they give the ICV injection. Then injection is mainly a choroid plexus cells. Then you will see that this is a control. It's artificial cerebrospinal fluid. Then you will see different kind of cells. The BRDU is a very unique marker to mark the newly produced cells. Anything that is new, and the BRDU can incorporate into the DNA. So you will see those are the new cells here. And with the choroid plexus cell inject into the lateral ventricle, then you will find that the subventricular cells actually become very abundant. Okay, so what the fact we found is that the choroid plexus can secrete certain factors. We don't know what it is, but those factors can upregulate SVZ, the neural stem cells activity. Okay, the next question comes what moiety actually carry the signals. You found that the choroid plexus does indeed modulate the SVZ. So what is the stuff that in the choroid plexus that modulate or carry out the signal from the choroid plexus to the SVZ? Okay, so that's what did the next stage we did. Actually, we tried to find out the vehicle. Now, why we are not focused on the certain factors we know of? There are so many factors that are secreted by the choroid plexus. But we believe that the choroid plexus has ability to secrete the small extracellular vesicle that actually contain or the package a lot of different kind of factors at once. Because in order to regulate adult neurogenesis, one single factor may not work alone. It must be something else that combined, including the metals as well. So that's what we did, actually try to find out the vehicle, especially for the long distance traveling from the choroid plexus to the subventricular zone. Anatomy speaking is quite close, but within the cells is considered as a long distance. It's not like a presynaptic neuron to the postsynaptic neuron, several microns. So this effort we use is to identify the small extracellular vesicles. Okay, then try to looking for the evidence for those small extracellular vesicles as a carrier molecule. So give you the concept about an exoson. Exoson is part of a small extracellular vesicle. Okay, it's a part of it. So the exosons that there are four basic criterions. They must be the small, of course. Usually is the less than 200 micron, and they can carry the cellular cargoes. It's not just the one single molecule. It has a DNA, proteins, and lipids, and a lot of signal, very unique signals that are produced by the choroid plexus. It must be able to travel to the certain distance, okay, and also can do the signal transduction. That's a basic concept. So what we did first is try to see if there is a marker for the small extracellular vesicles. There are different kind of the markers. CD63 is the most common one. You can see this, all those markers for the small vesicles expressed in the brain, that the choroid plexus is much, much higher as compared to all the rest of the tissue. SVZ also have those kind of molecules, but the choroid plexus has a much higher. In other words, choroid plexus as a brain tissue, also the size is small, so very actively produce the extracellular vesicles and sends the signal to the rest of the brain through the cerebrospinal fluid, of course. And you will also see those the markers that in the choroid plexus tissue. If you take the tissue out and to do the immunocytochemistry, you will find out those are the markers that's all in the choroid plexus. Indeed, what we found is the choroid plexus does possess the small extracellular vesicle, and also the amount of the small extracellular vesicle in the choroid plexus is far more abundant than in the other brain regions. Okay. Now, next question is, can we actually purify of this small extracellular vesicle and characterize it? Okay, so that's what we did. Actually, the students spent about at least the 4 or 5 months try to use the centrifugation method to collect the purified small extracellular vesicle. Then once you got the purified small extracellular vesicle, then you incubate with the neurosphere. Remember the neurosphere that we produce? We used to incubate the neurosphere with the choroid plexus. Now we take the choroid plexus out, just incubate with a small extracellular vesicle. So that's what you found. Actually, once you incubate with the small extracellular vesicle, not the choroid plexus tissue, you found that the neurosphere grew bigger, and also have the different kind of a cell differentiation, migration, and the reproduction. So you will find out the choroid plexus secret small extracellular vesicles are critical mediators for the enriched SVZ neurospheres growth. So this is the conclusion we got, that actually the choroid plexus indeed produce the small extracellular vesicle, and those vesicles help to stimulate adult neurogenesis. Now, then we further actually compared small extracellular vesicles, that the preparation, and check for those markers. You will find out this is the purified, relatively purified SEV express those markers much bigger much higher as compared to the choroid plexus cell lysate. Then we use the electron microscope. You found that those particles, and the fifth with a concept that is established by the International Society for the Extracellular Vesicles. There is a standard. And also we did the size distribution analysis, then found out that most of them actually around the 100 to the 170. In general, it's less than 175 the micron. So that's fits the concept. Choroid plexus indeed produce the small vesicles. One is from the functional wise speaking, that if we incubate and it actually stimulates SVZ and the neural growth, and physically you will find out that indeed is the small vesicles secreted by the choroid plexus. Next goes a little bit complicated. So we put the small vesicles into the tubes we call the pump. Okay, this pump can slowly release the small vesicles. So you put the pump actually into the animal on the back. Okay, so that takes a while to slowly actually release the small vesicle into the brain. Then we like to see that if that affect adult neurogenesis. Okay, so you look at this one, you have the PVS setting, and with the controlled the small extracellular vesicle. Okay, this one you found that you will find the BRDU express cell is significantly increased. Those are the arrow point out of this. And now this group is the manganese treated as the negative control. It's the manganese toxic compound accumulated in the choroid plexus. So we believe that the manganese exposure can alter the copper metabolism in the SVZ. Okay, so we use the manganese as a kind of the control. So you will see this once you count it. BRDU labeled the neurons. After you infuse into the small extracellular vesicle, for about 5 days, you see that the new neurons significantly increase. Whereas the animal treated with manganese, you will find out that this is a significant decrease. So in other words, that the choroid plexus secret small vesicles modulates adult neurogenesis in SVZ. So this is the other line of the evidence. Okay. Now, we also have found out, because from the pharmacology point of view, you can always inhibit something, stimulate something, use the antagonize, and also the use the agonism, right? But this is we use the more the recent technique, the gene regulation, to knock out the specific proteins. And this one is the SMPD3 that has been known in the literature that can catalyze the biosynthesis and release of small extracellular vesicles. It's not from the choroid plexus, but from a lot of study from somewhere else specifically demonstrate this is a very unique protein. That protein can modulate small extracellular vesicle that synthesis and release. So our idea is that can we knock out or knock down this SMPD3 in the choroid plexus and see what happened in SVZ? Okay, so this is a technique we including the gene modulation. So what we did is that you have this AAV5 that is vector, and with this vector you actually have the short hairpin that the RNA. You design this RNA, just the SMPD3, specifically hit on the SMPD3 so that the protein production, the DNA, it will not affect at the DNA level. So it's not a genetic modulation, but rather at the protein level. You can control the protein expression of the SMPD3. Okay, reduce the production of this, see what happened in the SVZ. So this experiment we did, it's a very tedious experiment. It takes a long time, and you fail once. Actually, the 5 months that pass, they have to start again. This is really the painful experiment. The student didn't like it, but eventually worked out. So give the AAV5 and the labeled that is the vectors inject into the animal. You found out actually with the silence of the RNA indeed reduce the SMPD3 expression in the choroid plexus. Okay, then you found that this slide really shows you AAV5 is a very unique vectors can recognize the choroid plexus. Not all the vectors can recognize the certain tissue. Okay, once you inject any the vectors could affect different places, but the AAV5 is very unique, can recognize the choroid plexus. You see this is not on the brain ventricles, it's not in the SVZ, but specifically in the choroid plexus. And this slide shows you quantitatively speaking that after you inject the AAV5, then you found out the expression of the SMPD3 significantly reduced. Okay, this is another study. The shortened effect, the 3 months post-IAV injection, and we gave the BRDU. BRDU is labeled the newly produced cells. Then you dissect animal. You will find out that after the SMPD3 injection, the cells was significantly reduced. The BRDU that labeled the cells in SVE was significantly reduced, and so is here. Okay, so again, this is evidence suggests that the suppression of small extracellular vesicle secretion from the choroid plexus. I mean, the vectors recognize the choroid plexus, but the end point is in the subventricular zone. Adult neurogenesis reduced. Okay, so in other words, you see this, we talk about the evil axis. So you have a choroid plexus, you have SVZ. So you inhibit this one and actually affect on the end point. Okay. Now, the other question is that, once the SVZ got damaged, and SV supplies a lot of neurons eventually to the olfactory bulb. And we know the olfactory bulb control the smell function, right? So does that affect the smell functions? So this is a kind of a functional analysis. You give the injection, that's the suppression SVD secretion, okay? Then you with the mice that take the food out, let them to be hungry for a while. So you hidden the food pellet under the bedding system, and the ones that are hungry, they start to looking for the food. Okay, this is normal control, and they use this one actually the knock down the SMPD3 in those mice. Those mice, it takes a longer time to find the food. It's because it's not saying that they are not hungry. They are hungry, but it takes a longer time to find the food. It's simply is due to the damage to the SVZ, damage to the smell functions. So this is a first experiment. 6 months later, you will find that that animal actually takes even longer time to find the food. And the second experiment that related to the smell is that we can actually, the male animals tend to smell the female urine. And once you put the female urine on the filter paper, the male animal will stay longer time. Normally, they stay longer time with that urine because of smell function. If you damage the smell function, the animal may lose the interest on just couldn't they smell. So this is experiment we did. We did the study with the filter paper. You drop some of the ceiling, then the test for the 2 minutes, rest for 1 minute, test a second time, test 3 times. You will see the animal gradually lose the interest. Now, at this point, you add the female urine on top of this filter paper. Actually, you will find out that that animal stay with the control animal stay longer time, 12 seconds, whereas the SVZ knockout got a damaged animal. They spend much less time because it gradually lose the function. And the same thing here. At 6 months later, that animal will stay, the normal animal stay about 12 seconds with this female urine. But this with the smell function got damaged. Actually, they are not interested. It's only 5 seconds with that urine. So that means that you give the knockdown that the protein modulates the SVZ or modulates the choroid plexus production of small extracellular vesicles. Eventually, is going to affect the smell. And one more experiment we did is the avoidance test. You give the 2MP to have the smell so bad, so the animal actually running away with this. Okay, so you look at this. A lot of animals actually are running away with this, but with the SVZ damaged animal actually stay longer time with this smell things. Okay, so those experiments all tell the one thing. That is if you damage the choroid plexus, the function to secrete that small extracellular vesicle, the end point is that it will affect the smell functions. Okay, so you have the two ends actually linked. So the question is that is there a choroid plexus with a regulatory axis, or we call the EVO axis? We believe yes. Okay, there is the axis. Is quite important and the transport made it through the cerebral spinal fluid. And why I mention cerebral spinal fluid? Because this is the place, the median, our clinician can collect, analyze, and rationalize, then compare with something. So if in the disease condition, and you got a cerebral spinal fluid, we'll be able to measure the small extracellular vesicles. Depends on what the disease stage. Maybe we can use the small extracellular vesicle as kind of the biomarkers for the disease progression. Okay, so this is a study we did, and we found that there is that new concept. We believe there is the epithelial ventricular interactive lineage axis. We call the EVO axis, and students love it because it's easy to say this. Okay, now why we have another name called the CSR axis, because the lineage is really implied. The start point is really to see how that affects SVZ. Generally, it's a different number of new neurons. So that's related to lineage because of cell differentiation, cell production. But is that the end of it? Is the SVZ is only doing this, or the SVZ has some other functions? So the more broadly speaking, probably the CSR axis can embrace a lot more concept. So far, we don't know yet. Okay, so this is here actually that shows you that is how that the axis works. Choroid plexus secretes the small extracellular vesicle, and this vesicles attack the subventricular zone, and eventually affect adult neurogenesis. Here we talk about adult neurogenesis. Then we talk about Alzheimer's disease. Okay, we talk about some other disease. You need to have some mechanism to compensate whenever the cell loss. So we talk about the cell repair process, neuronal repair process. Does the choroid plexus eventually contribute to the neural repair process? So that is a very important thing, and I believe for next many years, this could be if we have more people involved on this concept, we can better understand if there is the axis and how that axis really works. I also like to mention that the metals stored in the choroid plexus can disrupt the normal small extracellular functions, especially in our manganese study. Today, I don't have time to talk about this. And also, we believe that as the small extracellular vesicle can serve as the potential therapeutic carrier for the treatment as well. Now, I'm going to switch the gear to talk a little bit about the ALS lead exposure, and mostly I think I will leave the concept of ALS to the Roos. Professor Roos has a lot of experience, but I will talk about lead exposure a little bit. So for the ALS, one of the major things, as Jan just pointed out, that is about the neuronal, the loss of the neurons in the central and in the peripheral as well. So it's a progressive loss of neurons, not just in the neurons, but it's also the neuron, the cell actually matting sheets that surround these neurons, or the oligodendrocytes. That's the other thing. Now, where those oligodendrocytes coming from? Okay, the oligodendrocytes derived from the subventricular zone. Subventricular zone provide the oligodendrocytes, provide the new neurons, and they spread to the rest of the brain. So that's actually bring us to the concept that the oligodendrocytes is a consistent replenishment with new neurons derived from oligodendrocyte progenitor cells. Okay, so there are papers. There's a very good review article several years ago by the group of people. They mentioned that for the dysfunction of the ALS is related to the degeneration. So beyond the well-known cytotoxic protein aggressions and the DNA alterations and the neuroinflammatory reactions of mitochondria, there are two areas actually related to the small extracellular vesicle or the choroid plexus. One is that they mentioned there is impaired mRNA processing in the oligodendrocytes. That is related to the small extracellular vesicle. Remember, I mentioned that the vesicle carries the mRNAs, okay, and also alter oligodendrocyte differentiation regulators. That is also related to what we mentioned, that the EVO axis, okay. More recently, there is a paper about ALS and the extracellular exosome disruption. Then this paper even mentioned that the ALS, you can use different kind of the markers as the biomarker for the disease progression, but they have a very specific markers. It's not extracellular vesicle in general. So you can imagine the plasma, CSF, and the serial. So can we use the small extracellular vesicle as a biomarker? I believe we got to try this. Okay. Then this obviously is a very important things about the metal exposure. If there's a linkage between the lead exposure with ALS, so a lot of people think that the lead exposure is old story ever since the 1965 or 1968, the Needleman, Dr. Needleman at Johns Hopkins did a human study. He mentioned that lead actually is related to IQ changes among the children. Then the big story, somebody say yes, somebody say no. And the argument has been over there for about 20 years. But now, based on the data we have, the epidemiology data, not just in the US, but in the Asia, China, Japan, and also in Europe. I think some papers from the Scandinavia countries as well, that the lead-induced IQ change is indeed there. Okay, the lead can induce the IQ changes. So you look at it, the people will think, well, we already removed the lead from the gas ring, so why you worry about it? Okay, in New York City, there are a lot of lead pipes. So you already replace those lead pipes. So why you worry about the lead exposure? Okay, why we worry about it? It's because by the capitalism, you will find out if somebody pays a price to buy the particular stuff, that means that metal actually worth something. So you look at the lead a long time ago, is about $800 per ton. 2005, 2008, the prices increased. Anybody remember 2008? 2008 is the financial crisis. Okay, so in the world, all the government poured money into the basic constructions to rebuild the buildings, the highways. So lead consumption was increased. The prices increased. After that, start from 2010. Become relatively stable. So lead price has a very stable. It's not up, not down. So very stable. In other words, modern industry requires the lead. Okay, modern industry is not saying that we can phase lead out. We cannot phase lead out. You look at it. Okay, so here is a battery. We use lead with the sheaths and the lead using the weapons, and also use a lot of different things. So lead essentially required by the modern industry. So the exposure is still there. And just several weeks ago, I think this is last month, New York Times, there is a report that 200 kids hospitalized. Is it because in the kindergarten, they use the dye to color the cakes. Then the kids like to eat the colorful cakes that eventually got lead poisoning, and there is a cover up over there as well. Okay, now this slide I can surely show you what is the lead exposure level. Less than 10 microgram per deciliter is made in the children. We think that is the cause IQ changes. Then between the 10 to the 50s is usually we see in occupational exposure. And in some cases, lead concentration extremely high. You will see that the deaths around 150 microgram per deciliter. So here I show you a case that with a lead exposure. This woman, I think is 27 years old, is really healthy, but has some issues with the bleeding during the menstrual cycle. So instead of the lady looking for the clinical doctors in the hospital, she actually went to the traditional Chinese medicine doctor. Then that doctor prescribed the drug. It's called Sanqi powder. Sanqi powder, if you learn the Chinese medicine, you know this. Sanqi powder is a kind of make the blood running through smoothly. Okay, now physically the Sanqi powder is very similar to the lead powder. lead powder. Okay, so she went to the pharmacy, the Chinese pharmacy. So pharmacy gave her a bag of the senchi powder. So he took that senchi powder twice a day. Eventually got a serious problem that she has the problems that severe nausea, and couldn't actually stand it up, actually rolling on the floor. Then the doctors over there sent into the ER. So the ER doctor gave her different kind of treatment, but nothing can actually control this. Eventually sent over to the Beijing Caoyang Hospital, and Caoyang Hospital, I have the very good collaboration over there. So I bought tickets and I went over there, and the with doctors and over there to check, and with this is an interesting case. There are another case as well. So with EDTA treatment, eventually they bring the lead concentration down. Then this person then eventually was released. Oh, and everything's fine because she's very healthy, she's young, and they cannot deal with this. So this just tell you that clinical speaking, this is lead powder. She took the lead powder instead of the senchi powder. So that things indeed happen is a clinical error. So it's accident. So what I'm telling this is that the lead exposure is not just due to the environmental exposure or the occupational exposure. Clinically, you will see that the lead exposure as well. Okay, there are a lot of chance actually that people are exposed to the lead. So we did some study actually in the manufacturer smelting that manufacturer. You see the worker put the monitors over here. The lines goes all the way to here, so the monitor the air concentration of the metals. Then we also did Dr. Pira from Italy, is an occupational physician. So I invite him to go with me and to do the study of the check for the patients, and eventually we did MRI machines over there. Okay, so what we found actually with lead increase, so the concentration increase, you will see that the brain, the hippocampal volume was significantly decreased. So lead is indeed can cause the problem with the human brain, and how that the hippocampal volume decrease that related to the ALS, related to the multiple sclerosis, we don't know. But there's indeed with a lead exposure among those workers, smelters, and you will see that the brain structure changes. More recently, we did a study that found that the mice, if you give the lead a chronic exposure for 20 weeks, you did MRI study for that look at the marine sheets of the water in the marine sheets. MRI actually can pick up this sample. That study was conducted by Wei Zheng, that is the physicist and at Purdue University. He developed this methodology. So we found chronic lead exposure can cause a demarcation in corpus callosum. Okay, so remember the demarcation is a pattern of the ALS. So we think that the problem is related, and the most effective. I'm not probably go through the real quick. We remember we did the neurosphere assay. This is a neurosphere that coming from this coming from the mice. That the mice, you collect the neurosphere, then you will see that with exposure of the mice, that the neurosphere, different kind of a marker actually decrease. But once we incubate with normal choroid plexus tissue, you will see it actually reverses it. So the choroid plexus is like the medicine, can reverse the adult neurogenesis. In other words, I think the choroid plexus, this just tells how important the choroid plexus, this tissue is in terms of the adult neurogenesis. Okay, so there are a lot of other papers is about that exposure ever since the 1970s. Probably this is the first report that talk about the lead is related to the ALS. Then most recently, there is a study, actually 2019, that's about the correlation in Denmark, lead exposure and ALS in Denmark. I think that Dr. Roos is going to give the extensive discussion about this. So I will stop here for the lead exposure. Now, I'd like to really spend some time, a little bit more time. The last section is about the copper transport by the blood cerebrospinal fluid barrier in the choroid plexus. So is a copper is good metal? Yes, it's very necessary to the human growth. But on the other hand, copper is bad metals as well. It is a very active redox capability. Then it can generate free radicals. And a copper deficiency and the copper overload are all bad to the human's health. Okay, so our human body should have the dedicated balance or the control the system in order to keep the copper homeostasis is very stable. So here's the study we did the years ago about the copper the level in the manganese exposed smelting workers. And we went outside to the place to measure the blood concentration of the copper. You will find out that the copper concentration was significantly increased among the manganese exposed workers. Okay, and in other case, for the manganese exposed wilders in the Beijing area, and we also that the copper concentration, see that there is a concentration in the saliva, that concentration was significantly increased. Okay, and in the plasma as well. In other words, you have manganese exposed workers, you will see the copper concentration actually changes. So the copper changes that will affect a lot of things. So what actually the copper is in the body or in the brain? Is the copper free or the copper bound to something else? In general, the people believe that the 80 to 95% copper is covalently bound to the cytoplasmin. Okay, that has been demonstrated. And also about the small portion of copper is loosely bound to the albumin. All the rest, the free copper is a pretty small amount. But once we did the choroid plexus uptake, you have the choroid plexus tissue, you give the animal injection of the radioisotopes, give the IV injection or inject into the brain, then you dissect brain, take the tissue out, analyze the copper levels in the different places. You found out that the choroid plexus takes up lots of the copper, and mainly in the free copper is not in the bound state. Okay, if you give the copper albumin, you will find out that the choroid plexus will pick up about 1.5 to 2.0 in that range. Then if you give the copper cytoplasmin, even less amount of the copper will get into the brain. In other words, the brain transport seems to us is mainly mediated by the free copper. Also, the free copper is pretty small, but that free copper is a majority actually transported by the brain. Okay, here shows the same thing. The copper, the choroid plexus, and as compared to the rest of the brain. Okay, so the free copper is a primary species to pass across the blood-brain barrier and into the brain parenchyma and the cerebrospinal fluid. That's what we found. And also, this is the other study. We measured the kinetic of the copper, the uptake. Okay, so you will find out the choroid plexus is 1,000 times compared to the cerebrospinal fluid. So the choroid plexus control the copper, and it's a unidirectional transport, free coppers. Okay, and here is a frontal cortex, the striatum area. We measure it's all in the same magnitude, but the choroid plexus is way high in terms of transport of free coppers. So your choroid plexus, if you found out the infusion of the copper to the brain, copper ions accumulate in the brain tissue. Choroid plexus is much higher as compared to the brain, the capillary or the blood-brain barrier, and much higher than the parenchyma and then in the cerebrospinal fluid. We published a paper years ago. Now, the other study we did is culture the cells, the mimic the blood-brain barrier. So you have broad cerebrospinal fluid barrier. Here in the middle is a primary choroidal epithelial cells. It form the monolayer. Now, then you have the inner chamber, outer chamber that is separated by this broad cerebrospinal fluid barrier. Now, we add the copper into the inner chamber, measure the copper radioisotope from the outer chamber. That way, we can measure so-called efflux, or we add the copper into the outer chamber that is facing the blood, then measure the copper concentration radioisotope in the cerebrospinal fluid. This is cerebrospinal fluid. So this is what we call the influx, and we compare the both, which way the choroid plexus prefer to transport. So turns out actually copper, and we use a space marker, you found out the efflux is predominant as compared to the influx. Okay, in other words, the choroid plexus pick up the copper from the cerebrospinal fluid, transport from the cerebrospinal fluid to the blood. So the choroid plexus, the function is the efflux is predominant inside of the brain. So we also try to see what kind of a protein or transporters in the choroid plexus. There are different kind of transporter. One is called the CTR1, the copper transport protein 1, DMT1, divalent metal transporter, ATP7A and ATP7B. You found out actually compare the brain, the parenchyma, and the choroid plexus, the tissue. Choroid plexus is solid. In general, is pretty high. This is the CTR1 and DMT1 is almost the same level. ATP7A is a primary in the choroid plexus. ATP7B is a primary in the blood-brain barrier. So blood-brain barrier, cerebrospinal fluid barrier, that different capacity for the export. In the this ATP7A is made for the efflux, so it's mediated by the ATP7A, whereas in the blood-brain barrier, endothelial cells is mediated by the ATP7B. Now, those two, CTR1, DMT1 is all the same. So the argument is that do they play the same role to transport the copper? So we did another study. It's a very tedious study as well. So we create the cell type is called the tetracycline inducible CTR1 or the DMT1 cell lines. In other words, you have this probe inside of the vectors. Once you add the tetracycline, add into the culture medium, okay, then the lock actually unlocked. So cells start to produce excess amount of the CTR1. So this slide that shows you, once you add the tetracycline, the overexpressed of the CTR1. So we want to see if the cell is inducible those transporter, what is going to happen to the metal uptake. So in the CTR1, you will see the CTR1 expression that increase once you add into the tetracycline expression, the CTR1 is increase. And here's the DMT1, the increase as well. But for the copper uptake, once you induce the transporter, uptake is an increase as well. Okay. Now look at the scales. Here's a 2000. Here's a 100. Okay, it's a difference. You will find out CTR1 is much more actively transported the copper. Okay, as compared to the DMT1. So we think that at the broad cerebrospinal fluid barrier, at least this CTR1 copper transport protein plays a major role. So here is a summary that we have. And once in the lifetime, okay, here is the choroid plexus, microvilla on the top. The other side is the blood. Microvilla is facing the cerebrospinal fluid. We have the CTR1 and we have DMT1. Copper is transported as primary by the CTR1, big arrow, and goes inside of the cells. Then inside of the cells, ATOX1 actually bound to the copper, transported the other side. This is what we call the copper chaperone inside of the cells. And it is made at the above cells of a barrier is made transport by the ATP7A. Then there is ATP7B, small amount actually transported back to the cerebrospinal fluid. And inside of the cells, a lot of copper actually bound to the metallothionein. we did find the metallothionein level is very high in the choroid plexus. So this is the whole picture in terms of the choroid plexus transport. I'm talking about here is pretty easy, but it's about 5 to 7 years of work. We wrote the review articles. Actually, the Andy Mano was the one primarily working on this one for his PhD thesis. So most recently, we tried to measure the metal distribution with the specific instrument. It's called 3D synchrotron X-ray fluorescence imaging technique. That equipment that is close to Chicago in the National Laboratory. So you put the neutron, okay, the accessory of the neutron to the very high speed eventually hit on the target. On the target, you can see the spectrum. That one measurement, you can measure about 7 to 8 different kinds of metals. Now, what we found, astonishingly, that there's a very high copper level in the brain. That is the subventricular zone. This is unexpected, because what we want to see is a metal distribution in the hippocampus, okay, in the different brain area. But it turns out that the copper level is so high here. That is the one we start wondering, what is this place? Like the such a high amount of the copper. Okay, so and we found out that the concentration in the SVZ for this copper is about 18 to 65 milligram, whereas in the cerebrospinal fluid, 0.5 micromolar. Concentration-wise, you have the 1,000 times the difference. And eventually found out this SVZ that is generates the new neurons. So this is the finding actually the push our lab into the SVZ area. And not only that, we found out that this lab, the copper concentration in SVZ is increased with age. In other words, when I was young, probably the copper in my brain is not that much in SVZ. But with the age increase, the copper becomes higher and higher. The higher copper, we believe the hypothesis is a high copper actually suppress the adult neurogenesis. Our neuron repair the mechanism actually due to the high copper accumulates there. So it's the ability actually to decrease. Okay, so we think that the Scarhart disease is age-related, right? And this disease in the Norway is age-related. Is a copper ion involved, and also with the white matter lesion. In other words, is a degeneration. So we think that with that thing, that in general you have a concept is age-related, is a copper-related. So there is a good chance that the subventricular zone, or what we call the evil axis, may somehow contribute to the disease progression. This is just a hypothesis. Okay, so maybe somehow the related. So here is my final thought. I still have 5 minutes. So we think there exists the CSR axis or the evil axis in brain that regulates the neurodegeneration, the neural repair process that essentially to disease onset or the progress of ALS. And also we believe that the copper accumulated in the choroid plexus may somehow impair the function of the CSR axis or the evil axis, cause the damage. And the efflux of copper from the cerebrospinal fluid is regulated by the choroid plexus. That is the solid fact we found from the laboratory animals. And we believe that dysfunctional choroid plexus in the copper regulation due to genetic disorders or the environmental exposure may somehow contribute to the high copper level in the cerebrospinal fluid. I don't know if that's related to Scarhart disease, but probably would be at least partly may contribute to the disease, the causation or for the disease progression. Okay, so finally, it's just a question marker. We probably have a lot more work to do to demonstrate this. Finally, I like to give my deeper appreciation for the colleagues that help us, the Mark Siegel and Rashidin in the UK, and also Jean-Francois and Ariane project. He was the one who gave us the AAV5 that secrets about the AAV5, the vectors. He knows a lot about this, so I learned this from him. And also have the collaboration in China. Of course, they did the few works for us. And neurology in the department of the Indiana University School of Medicine. Then the Brad Grasset have a great interest in the lead exposure. So we collaborate with the project funded until 2026. And we have a physics, Julia Poshka at Purdue, give us a lot of help. Then Luke Liu, now at the Stanford University as a postdoc, and he is the one mainly did the EVO Access project. And over years, we have a lot of the students work through this. For the copper research, and Andy Mano is the one did a lot of study, and along with Dr. Su Zhang and the Sana Ho, and she graduated from the PhD later, and in some other lab, and also Shirley Anderson. So I really appreciate this. The student and the scholars made a contribution to the lab research. I'm the one actually give a talk, but they did all the hard work. And finally, I really enjoy this. The Oslo is a wonderful city, wonderful opera house, and a wonderful operas over there too. So we really enjoyed, and thank you so much for the invitation. Thank you.
Other questions from the auditory? It is complicated with this subventricular zone and so on. One question from Per.
If it's okay.
Yeah.
So the subventricular zone is very interesting. But I wonder how much evidence is there that it is important in humans as well? We know it's very important in animals, but what do we know?
Yeah, an SVZ actually was the first known from the human. Because the cell culture system is kind of difficult to dissect, so they normally start with the big animal. Now, with the time passing by, the technique become the more sophisticated. And the SVZ, the function to produce the new neurons, that has been very well known. And in the Alzheimer's disease, there are two areas that actually neuronal cell loss and also with the functional change of the neurogenesis. I think that has been known. But there is argument, some human studies saying that with Alzheimer's disease, SVZ, the new neuron actually decreased. But some other study actually say SVZ neuroactivity increased. So the argument is that with the cell loss in the hippocampus, there's a demand for the brain to produce more neurons. So that's why you see the SVZ, the neural production, become very active. So this is a human study that has been very well known. And in the animal model, in our study, we haven't actually the check in the Alzheimer's, the animal model, does the SVZ function change? We haven't done that study as yet. So it could be the good project for some graduate student.
In Skogholt, we found these very high levels of both copper and iron in cerebrospinal fluid. Apparently, there is a metal transporter that does not function. You think it is an ATP requiring?
Yeah. The copper transport obviously is driven by the ATP. ATP7A or ATP7B, but we think that in the choroid plexus is a primary ATP7A. So there's also the other comments is that the copper and the zinc is always thinking about the zinc is a tree is the opposite to the copper, is a good metal, but the copper itself, if you look at the cellular biology, copper is quite toxic, and iron too. So you add the copper and iron together, I think that the addictive effect, I don't know if there is one, but if there is a copper and the iron both present in the cells, I think they can cause the more damage to the neurons.
I think we will come back to the myelin
Yeah.
brain sheet in
Yes.
the next
Yes, myelin sheet.
presentation,
Yeah, myelin
and
sheet.
you can comment it.
Yeah. And also, I like to comment that chelation therapy, Jan is the pioneer in this area. I know your name actually is started from the chelation therapy, a book like this, that many, many years ago when I started my career. So thank you. It's really the privilege to speak here and share the stage with you. Thank you.
Thank you very much. We had a question from Jan Alexander.
Thank you for a very interesting lecture. I have a question about how do these small vesicles reach the subventricular zone? How do they pass through the first cell layer that's covering the wall?
We don't know what is the exactly mechanism that a small extracellular vesicle, for example, is secreted by the choroid plexus around, and they arrive the targeted area in the SVZ. SVZ is pretty long, so it's not a small area. It's not like linear. Actually, it's a pretty long area. We don't know where this small extracellular vesicle landed, what is the target, and what's the inside of the small extracellular vesicle that play the critical role. So this the EVO axis actually brings a lot of questions than the answer. That opens the doors for probably a lot of graduate students to do the study. There is a lot of unanswered questions. I think the one you mentioned is it is critical.
Yes. Thank you again. I think we should go over to our short break. Thank you, Professor Wei Zheng. And we will be back here at 6 o'clock.
Thank you.
I see. This rare disease we have already discussed. It is probably not a kind of Alzheimer. It is a very special disease. And the next speaker here is senior researcher Per Selnes. More about the choroid disease.
Okay, good. So thanks for having me. No, this is not a kind of Alzheimer's disease, but it's, well, in my mind, incredibly important to understand Alzheimer's disease and also understand how the choroid plexus works. But to give you a bit of a background, so this is a journey from several decades of hypothesis to what we argue is a mitochondrial choroid plexopathy. So there have been shifting hypotheses for the cause of this disease. Demyelination, poisoning, is it that tauopathy or is it dysfunction of the blood-brain barrier? And this talk today is based on what is known as Skogholt's disease. And for those of you who are non-Norwegian speakers, Skogholt is an eponym. It's a name, but the name itself, the word itself, means either a copse or a thicket. So that's just a curiosity. Good to know. But this publication in 2023 that I took part of is what inspired what we see today. That is sort of the basis of the understanding that I hope I can convince you through today's talk. So today, what you will hear is from an unpublished manuscript, just pending genetic confirmation. So we are sequencing genes to sort of pinpoint the mutation and hopefully sort of get all the pieces of the puzzle. But there is everything culminates in a unifying explanation of a mitochondrial choroid plexopathy, and I will try to take you on the path from strange hypothesis to a coherent disease mechanism. So this is what we know from the 2023 publication and from previously. So we have a very unusual finding of very high protein levels in the CSF. It should be blank, like hopefully most of our CSFs are, but in these patients you have a discolored CSF that has very, very high levels of proteins, amyloid beta, t-tau, all the biomarkers you can imagine are extremely the concentrations are extremely high in the CSF. And late stage, you get these white matter hyperintensities on FLAIR MRIs. That is usually attributed to small vessel disease of the brain. In the end, I hope I will have convinced you that this is not due to vascular disease. It's something entirely different. And what we also need to remember is that although the CSF looks like this, the levels in plasma are either low or normal in these patients. So that's some major clues right there. But some more work was needed. So this disease exists only in one family line in Innlandet. It's in rural Norway. And the inheritance profile was not quite that known. So, but I tried to draw the pedigree this Easter actually, and it looks like this. So this is a truncated version of the pedigree. So it's a Norwegian multi-generational kindred, starting with 9 siblings who are all affected by the disease. And it becomes apparent when I drew the new pedigree that it's strict maternal inheritance, and that is mitochondrial etiology until proven otherwise. So, and the patients get very early fatigue in their late teens or early 20s. They are very fatigued, but they have no other overt symptoms. But later they develop these subtle neurological signs that wax and wane, and they get these white matter hyperintensities that we saw. So you have this long smoldering course over decades, and when we see this family tree, this pedigree, it's mitochondrial. It has to be. So we are sequencing mitochondrial DNA right now with long read in collaboration with Newcastle, but the results are not ready yet. Okay, so why the choroid plexus? Why do we think that the choroid plexus here is important? So in the 2023 publication, we saw some signs that the choroid plexus was small. I didn't really trust that because we had so many different scanners, and there was the scanner drift, and you have to correct for scanner. So, but there was at least an indication. But what we know is the choroid plexus is the main producer of CSF. So it drives turnover and solute clearance. And it does that in 2 ways. So you have the bulk effect of CSF, that is sort of the engine of the lymphatic system that cleans the CSF or cleans the brain of solutes and waste by pushing it through a perivascular pathway. And you need CSF for that because you need the pressure to push through this pathway. And additionally, you have ATP intensive transport hub. So you have all these transporters in the choroid plexus that use ATP, which is the energy of the cells derived from the mitochondria, that actively transport waste out of the brain. And also the production of CSF is extremely ATP intensive. So this issue is very mitochondria rich. It is actually the most mitochondria rich tissue in the brain, you can argue. And it also has to function 24/7. It can never rest. A muscle can rest. So if you have a mitochondrial disease, your muscles are strained, but they can rest. The choroid plexus can't rest. And additionally, it has a very low glycolytic reserve, so it can't use other types of energy. So you get a depletion of ATP in the choroid plexus, resulting in reactive oxygen species and, well, a toxic environment. And I argue that Skoghol's disease shows what happens when this system breaks first, that this is the cause of what we see further in the disease. So how can I say this? How can I be so sure? Okay, so we have some animations. I wasn't aware. Okay, but there is also a link to Alzheimer's disease here. So I think this is that is why I became interested in this disease in the first place, because it felt apparent to me that it has to be very important for Alzheimer's disease when you have this major buildup of biomarkers for neurodegeneration in the CSF. So you have like sky-high tau levels, which is at the same level as you see in prion disease, in Creutzfeldt-Jacobs disease. So it's sky-high, but it's not necessarily an apparent atrophy of the brains of these people. And in Alzheimer, you see accumulation of A beta and tau. And there is a growing recognition that clearance failure is a major driver of Alzheimer's disease. And in Skoghol's disease, I would argue that this is a human experiment of nature. You have a clearance failure without massive neuronal loss, and you have this plasma CSF dissociation that shows stagnation, not just degeneration. You have completely normal levels of biomarkers in plasma, but sky high in the CSF. So this provides a model to study upstream choroid plexus dysfunction. And that gives us lessons for AD. And I was obviously meaning to put a picture there, but I must have forgotten. That's just a reminder for myself. Okay, so let's go to the biomarker paradox. So this is, I needed to use a logarithmic scale here to get the changes here. So you see here, this is from a previous publication, and I reused the data. This is from the 2023 publication. And what we see here is actually you have a 10-fold increase in tau and 12-fold increase in phosphorylated tau, which are major Alzheimer's disease biomarkers. And you also have severely increased many other proteins. And beta trace protein, amyloid beta species, albumin, neurofibrillary light chain, NFL, and GFAP, and they are all very much increased in the CSF. But most of them are normal in plasma. But very interestingly, GFAP and NFL are low in plasma. So what does that tell us? Well, most of these other proteins, the amyloid beta and the taus, they are actively transported either across the blood-brain barrier or the blood-CSF barrier, from CSF into blood. These two, GFAP, which is glial fibrillary acidic protein, it's only produced in the brain by astrocytes, and NFL in health, it is the plasma, the NFL in plasma in health is more or less only brain-derived. But they are low. So that tells us they are not actually transported out of the brain. They depend on bulk flow. They depend on the glymphatic system. And that should tell us that the glymphatic system is not functioning due to decreased production of CSF. So these are trapped in the CSF, and therefore, because they have no external, no peripheral source, they are depleted in plasma, which is a piece of evidence that something is going on with the choroid plexus. So this implies a clearance failure, and it's not at all an explosive neurodegeneration. You see this pattern at a stage in disease where the patients are, well, they have their problems, but they're ambulant and they function well. So we repeated structural MRI. So this is a structural MRI sequence. And I did some deep learning procedures and to segment the different areas and functional areas of the brain. And so this is a plot. So this is effect size, Cohen's D. Usually, if it's more than 0.8 or something, it's a very large effect size. And this is significance. So this is minus log 10p, which is just an easy way to show. This would mean minus log 10p5 is 1.11111. Sorry, 0.00001 in p. I think I got the right number of zeros there. But the point is here, the choroid plexus is very small in this disease. It's a lot smaller than in the than in controls, and also the ventricle volume is a lot smaller in these patients. So you have a tiny choroid plexus in these patients, and you have small ventricles. And other changes in the brain, there are no major structural differences between patients and controls, but you have small ventricles and you have a tiny choroid plexus. Okay. And then I thought, okay, we have to check the choroid plexus in a different way. So DCE MRI is a dynamic contrast enhanced MRI technique in which you use small gadolinium chelates as a contrast agent, and then you take 100 T1 scans in like 5 to 10 minutes, and you see how perfusion is in the brain. But the problem is that the choroid plexus, when you analyze this type of MRI, all the assumptions are not met in the choroid plexus because the choroid plexus has active transport. It has fenestrated capillaries, so you can't really use the standard ways of measuring. So I just looked at washout. What is the washout of the contrast agent in the choroid plexus? And so this is time above half peak, and it has a Cohen's D of minus 4.9 and a huge significance. So this means that the differences between patients and controls are enormous. So there is an enormous difference in contrast washout. We don't really need statistics. We can just look at these curves. So what you see on the bottom here is controls. And this is what you would expect. You inject contrast, and then you get a huge peak and a washout. But this is the patients. Look at this person. The contrast is injected, and then you just trickles in and stays in the choroid plexus. And it becomes very apparent that this is a huge difference between patients and controls. And then next stage, I looked at other parts of the brain using this same technique. And here we used the traditional methods that is used with pharmacokinetic physics models to explain what happens in other parts of the brain. And yes, we also find changes in the choroid plexus, but it's difficult to interpret because the fenestrated capillaries and the very different milieu that is in the choroid plexus as opposed to the rest of the brain. But what we see is especially that a measure called Toft's K-trons, which is bidirectional flow of this contrast agent, gadolinium, it is very much lower in patients, which is a very, very unusual finding. What you usually see is it could be increased if it was a damage of the blood-brain barrier. You would expect an increase in K-trans and also in packet lack, which is unidirectional. so the point is here that you get a very different profile in the all over the brain of these patients than you would expect, and very, very strange that it's lower. That is not something you see in any disease. It usually increases with disease. And the only thing that could, well, in my mind, please come up with other explanations, but the only thing that could explain this is solute stagnation in the tissue, in the cortex and in the deep gray matter. You would then get, so if you have a protein stagnation, because the lymphatic system is not working, you would get proteins stagnated in the parenchyma, and you would get this pattern with reduced exchange of the tracer. But then we thought, okay, there is something else we could check. We also have diffusion tensor imaging on these people. And what you expect in Alzheimer's disease, you would expect a huge increase in mean diffusivity. So you would expect that diffusivity is increased with neurodegeneration. But you would also expect, if you have stagnation, you would expect a decrease in mean diffusivity. That is also something you don't see it in disease. That is not something that is reported in the literature. But the hypothesis was, okay, if this means that there is solute stagnation in the tissue, MD, mean diffusivity, should also be increased. And yes, it was. So all over the brain, there is a decrease in mean diffusivity in this disease. And so this is just an attempt to illustrate free diffusion. So everything can just diffuse freely, but if you have all these proteins and blockage, you have a stagnation, and you would get a decrease in diffusion. And the only exception we saw to this was the putamen. There we see an increased diffusivity, and that may sound strange, but in other mitochondrial diseases, it is a very common finding to see changes in the putamen first. So that also is sort of a clue that it's correct, that it is a mitochondrial disease. And then we have these trace metals that I suppose many of you are quite interested in. And this is also a reanalysis or reinterpretation of a previous publication. It's 2008 Glenn publication. And okay, so we have in Sjogren's disease, we have high, I selected 8 trace metals from the huge list. And I chose these because we know they have ATP-dependent export at the choroid plexus. I chose these 2 because we know they have ATP-dependent import at the choroid plexus, and these 2 because they are probably passive. There is no active or transport or import. So what we see here in all of these metals that are actively exported, they are increased in this disease. Those that are actively imported are decreased in the disease, and the ones that do not have ATP-dependent transport are unchanged. So this pattern, I would argue, can only be explained by an ATP-dependent transport failure. At least the pattern matches that. So I'll try to explain why the choroid plexus is the first to fail in a disease such as this with a putative mitochondrial mutation. So the choroid plexus is a fragile powerhouse. It's the mitochondria rich. It has hardly any glycolytic backup, and even a mild shortage of ATP would seriously collapse the secretion of cerebrospinal fluid. So there were these animal experiments in the 60s and 70s in which they injected an ATPase inhibitor into the ventricles of mice and rabbits. We saw a 70% 30% drop in CSF production, showcasing the fragility of this system and how very ATP dependent CSF production is. So this is my attempt at explaining what happens. So this is supposed to be a healthy choroid plexus. You have mitochondria that produces ATP, and these active transporters transport trace metals, biomarkers, waste out of the CSF, and they produce CSF. But in a mitochondrial choroid plexus dysfunction, ATP is not produced. There is a shortage of ATP, and these transporters do not work. So there is stagnation of the proteins and trace metals and waste products in the CSF, creating a toxic environment in which there is atrophy of the choroid plexus, atrophy of the blood vessels, and you have contrast agent trapped in the stroma of the choroid plexus. At least this is the proposed disease mechanism. So this is a rare variant, and the lessons for Alzheimer's disease, I touched on them. But in Skogård's disease, you have extreme CSF tau without neuron loss, and the interpretation of the AD biomarkers must also consider clearance failure. We know that there are changes in the choroid plexus in Alzheimer's disease, but if in Alzheimer's disease the choroid plexus cannot clear the CSF, how should we interpret these changes of biomarker levels? You need to look at clearance to interpret biomarkers in Alzheimer's disease, specifically in plasma, but also in the CSF. And this also opens a therapeutic axis if you can restore choroid plexus mitochondria and restore production of CSF. And also, it is a possible explanation for early fatigue in AD prodromes, which you often see. People that are in the early stages of Alzheimer's disease often get very fatigued and have pains and aches and anxiety. And that is exactly what these patients get up to decades before they get overt or clear symptoms. So the next steps is we're trying to go from one family to an organoid model. So as I started by saying, we're doing long read mtDNA sequencing to pinpoint the mutation that we believe is there. But that would sort of lock the mutation. I would guess it's complex 5. That is my best guess, but we won't know until we have the sequencing results. And then we are doing induced pluripotent stem cells, and we have already generated CHP-like cells from these stem cells and are working on doing organoid models. And so this is one of our collaborators, and this is not organoids from this family, this disease, but this is actually choroid plexus in a dish that is created from stem cells. And we're trying to do this now with cells from this family, with stem cells from this family, so that we can test what kind of modulators for mitochondria we can use to improve function of the choroid plexus. So here you have clearance first, degeneration second. So these patients get neurodegeneration later on, but what starts it is that clearance is not working. So there is convergent evidence here, although nothing is certain, but there are at least many things pointing to that this is a mitochondrial choroid plexopathy, and it provides a human evidence for dysfunction of the choroid plexus and a decreased production of CSF, which leads to these symptoms. And the lessons for Alzheimer's disease here is that clearance failure, they can mimic degeneration biomarkers. So it's not certain that the biomarker patterns you see in Alzheimer's disease is relevant for all, depending on how clearance works. We know that the choroid plexus in Alzheimer's disease in many patients is large. It grows with disease. And the size of the choroid plexus correlates with cognitive function in Alzheimer's disease. And mitochondria in the choroid plexus are a tractable therapeutic target that we can work on. So the take-home message here is that it's a choroid plexopathy. I've said that many times now. Clearance failure can mimic neurodegeneration biomarkers, and CHP mitochondria is a possible therapeutic target for Alzheimer's disease that we should and will work further on. And I don't think this is vascular disease, as you would guess if you are used to seeing MRIs like this. Everyone would think it's a massive small vessel disease in the brain. But in this case, I think this is stagnation of protein and waste products. And the proof of that is these other MRI markers that we checked that are only compatible with reduced or with protein stagnation, and also the fact that the CSF is already massively full with proteins and waste products that we can measure in the CSF. Yeah, I hope I haven't alienated myself too much from the general field of.
Thank you very much for the excellent. It was very nice to see you working on the organelle, the choroid organelle. So what are you going to do with this, if you successfully establish that organelle? So what's your next stage if you have these organelles? So what are you going to test out with these organelles?
We are, we have several lines of research going on, but so we will do CRS assays on the cells and see what kind of, how they respond to mitochondrial stress testing. And then we will measure the CSF that is produced because these are 3D, so we can actually check the CSF that is produced inside these organoids and compare between patients and controls. And we will, depending on the, I think is a complex 5 mutation. And then I've made a list of what kind of compounds we want to test if it is this and that type of mutation, and see whether we can rescue production of CSF. But this project is very young because it's only this Easter I realized it's the choroid plexus and it's mitochondria. So we're working on it, but we have a plan.
Question from Per Roos.
Excellent presentation. Thank you. Very detailed. I am caring for the patients, and I was thinking, did they deteriorate clinically after the exposure for gadolinium?
No. But so it's been a year since we added gadolinium. Would you think they deteriorated after gadolinium? What they do not want is a lumbar puncture. So we can get hardly anyone to do a new lumbar puncture. They say that they feel so bad for a long time afterwards, and that is usually not a problem. But I wouldn't think the gadolinium, because the gadolinium will not enter the CSF. So I would assume that this is not in any way harmful, more harmful for them than a normal patient to gadolinium. But please. And I'm also collaborating with a Dutch group in which we try to do ASL, which is a perfusion MRI that is non-invasive. You tag the blood and the neck, and then you use that as an internal contrast agent. And they claim they have a paper showing that they can actually measure flow of water over the choroid plexus, so they can measure the amount of CSF that is produced. So we're going to test that in these patients and in Alzheimer's disease patients. So maybe we don't need gadolinium.
Thank you again. We will have some discussion at the end of...
There's one more there, or should we wait?
Yes, we can take one more question.
Thank you. Maybe you said, but do they have lower pressure in the brain because of the lower volume of CSF?
Oh.
Does this affect the cognitive decline in some way? Because higher pressure is very bad. Lower pressure should probably do something as well.
Yeah.
Also, you mentioned they feel much worse than normal people about the lumbar puncture.
Yeah. So we have done lumbar puncture on a few of them, and it's been good. But we have lots of historic data, and this, I don't know how real that is, but they keep telling each other it was awful. I was bedridden for several months after lumbar puncture. From the historic data set. I don't know how true that is, but opening pressure is normal. It's normal to low, but it's within the reference range. And I think they don't have low pressure because I think this is something that's been going on since birth, since they were children. So therefore you get this very, very gradual change. And therefore you have these small ventricles. So I think that's the explanation why it's not pathologically low pressure in the brain.
Thank you. So we can now go over to the next speaker. Metallexponering ved ALS, metal exposure in ALS. Roos from Karolinska Institute. The floor is yours.
Tusen tack för denna invitation och möjligheten att snacka här i det norska akademiet. Tack till Aksel Bernhoft vid kommitté för geomedicin. Väldigt intressant att det är då just geomedicin som inviterar. Det är bra. I've done this in English language quite a few times in international conferences, but this will be the first time in Scandinavian language, och jag tänker att jag fortsätter att prata svenska som är mitt modersmål. Vem är det som står och pratar här? Jag heter Per Roos, jag är biokemist från början, Stockholms universitet. Jag är specialist i neurologi och klinisk neurofysiologi, utbildad i Uppsala i Sverige. Disputerade 2013 på en avhandling om just metaller vid ALS, Metals in Motor Neuron Disease. Och jag är docent i miljömedicin vid Karolinska Institutet. Detta om mig. Och temat är då metallexponering vid ALS. Jag kommer att prata om vilka metaller det handlar om. Jag kommer att beskriva yrkesexponering och titta på det här problemet från flera olika håll. Och sen säga någonting om vad som behöver göras för att försöka förstå vad det är som orsakar ALS. Och i vilken utsträckning exponering för metaller är inblandade i denna sjukdom. Som alla vet då så är det här en åkomma där huvudsymptomet är muskelatrofi och muskelsvaghet. Patienterna får talsvårigheter. De flesta har bevarat intellekt, även om frontallobssymptom är en del av bilden hos en del. Det är inga sensoriska symptom, det är en isolerat motorisk sjukdom, även om det har ifrågasatts lite här och där. Kardinalfyndet är fascikulationer. Vad är det? Det är små muskelryckningar under huden som kan ses kliniskt när man tittar på patienten, men också kan detekteras med elektromyografi i form av fascikulationspotentialer mycket tidigare i förloppet än man kan se det kliniskt. Så neurofysiologi och elektromyografi är nödvändigt för att sätta den här diagnosen. Sjukdomen går med denervation och den är alltid dödlig. Döden sker i andningssvikt, respiratoriska muskler förlamas och alla dör. Det vi ser här, det är pyramidbanan. Där kanske. Det röda du ser högst upp där, det är motorisk bark. Och det blå är pyramidbanan som degenererar vid ALS. Signalerna går från den motoriska barken ner genom pyramidbanan som är det blåa här. Förbi, i framhornen i ryggmärgen. Förbi den motoriska ändplattan, motor end plate. Ska vi se om vi kan få igång den här grejen. Du hjälper mig. Och det är alltså där vi ska köra. Framhornen, motoriska ändplattan, som ju inte är drabbad vid ALS. Det är ju en annan sjukdom, myastenia gravis, som inte är drabbad. Musklerna här ute verkar inte heller vara sjuka, förutom att de atrofierar då på grund av degeneration i på grund av degeneration i framhornen. Alltså degeneration och fascikulationer. Ja, vi börjar 1831. Ja. Den första beskrivningen av ALS är från 1831, en patientserie med 11 stycken patienter som har fått en oförklarlig svaghet. Och det är intressant här 150 år senare att konstatera att flera stycken i den här serien var just blyarbetare eller hade en exposition för bly. Den här bilden är något senare, 1862. Radcliffe sitter och tittar i sitt ljusmikroskop som ganska nyligen har blivit upptäckt. Och det här är ett tvärsnitt av en ryggmärg från en patient med ALS. Och vi ser friska framhornsceller och vi ser små degenererade tillbakabildade celler i framhornen. Och det ser ganska lika ut idag. Så 150 år senare så är det här en olöst forskningsgåta som har engagerat sinnet hos varje neurolog med självaktning på hela planeten. Där var och en har en egen idé om vad det är som orsakar det här. Så varför kan jag stå här då och påstå att det kanske har med metaller att göra? Ja, vi ska ta det lite stegvis. Incidensen av ALS har tredubblats på 50 år. Det här är svenska data och tillsammans med Gunnarsson, neurolog, epidemiolog, han och jag har satt samman från 3 olika studier incidensdata och det där är under review just nu så det kommer nog att gå att läsa om ett par månader. Och här kan vi se hur incidensen stadigt stiger från början på 60-talet och fram till någon gång 2012 eller någonting sånt där. Vad är det som orsakar den här stadiga ökningen år från år? Mest intressant är kanske den här dippen på slutet. På senare tid så har ökningstakten minskat och till och med minskat. Varför då? Hur ska man tolka det här? Kan det vara så att vi har sett toppen på ALS-epidemin då som den kallas och att det här är på väg ner nu? Det vore ju en lycklig utveckling. Eller är det så här att den här ändringen beror på regleringar i miljöexponering för 15-20 år sedan som har sin effekt nu? Den här kurvan är ett starkt argument för att vi har att göra med en miljökemiskt orsakad sjukdom och inte en primärt genetisk sjukdom eftersom de inte beter sig på det här sättet. Ja, jag jobbade här i Oslo på Ullevål sjukhus. Jag var neurofysiolog och tog emot hänvisningar. Jag tog emot remisser. där frågan var, är det här ALS? Och så utförde jag neurofysiologi och försökte besvara den frågan med ja eller nej. Och jag jobbade i många år på Ullevål med det. Men när patienten ändå var där då, så vände jag dem på sidan och gjorde lumbalpunktion. Och analyserade sen ryggmärgsvätska för metaller. Och det här är en sammanfattning av fynden. Små trianglar betyder metaller med neurotoxiska egenskaper. Runda cirklar är metaller utan sådana egenskaper. Det här är fallen av ALS och det här är kontrollerna. En standarddeviation över medelvärdet för de samlade koncentrationerna för de kombinerade fallen och kontrollerna innebär att man hamnar i den här översikten. Och de metaller som faller ut är aluminium, mangan, kadmium, bly och uran. Samtliga kända neurotoxiska metaller som förekommer i ryggmärgsvätska hos ALS, men inte hos kontrollerna. Ett intressant argument i riktning av att vi har att göra med exponering för neurotoxiska metaller när det gäller ALS. Möjligen ska man kanske sortera in koppar här bland de neurotoxiska. Vi har ju fått en fin exposé av koppartoxicitet av professor Zheng. Extra intressant är uran där koncentrationerna generellt var låga både hos fall och kontroller, men där uran fullständigt separerade fallen från kontrollerna. Vi hittade förhöjda uranhalter bara hos ALS-fallen. Så som har diskuterats tidigare, kanske är det en blandning här. Kanske är inte någon mer viktig än en annan metall. Men vi får se. Vi går vidare här och tittar hur det ser ut. Det här blev ju då min avhandling då, som är publicerad och går att läsa på Karolinska Institutets webbsida. Sen gjorde vi om det här nu. Alldeles nyss. Så att vi har upprepat samma sak och vi har studerat 15 stycken metaller, alla de isotoperna i ryggmärgsvätska hos ALS, och en lång historia kort så ser vi tydligt förhöjda koncentrationer av bly med en 3-stjärnig signifikans i ryggmärgsvätskan och förhöjda koncentrationer av selen. Så från de här data verkar det som att bly och selen är inblandade i pathogenesen ALS på ett eller annat sätt. Men titta här. Blyisotoperna är olika fördelade i ryggmärgsvätska, olika fördelade mellan ALS och kontroller. Och det visar sig att bly 208 i relation till bly 206 är förhöjd i ALS CSF. Isotopkvoter kan användas för att spåra bakåt varifrån exponeringen kommer. Därför är isotopkvoter viktiga och vi behöver upprepa det här på flera patienter och upprepa den här studien igen för att se om vi kan reproducera det här som vi nu publicerar för första gången. Ett annat sätt att spåra varifrån exponeringen kommer är att använda så kallade lantanoidmönster som ni alla vet i periodiska systemet. Lantanoiderna är 7-8 stycken där som har en speciellt fingeravtryck kan man säga. Man kan leta bakåt varifrån exponeringen kommer. Och för att få ordning på det här då så gjorde vi en metaanalys, en stor metaanalys. Vi är nu då Aida Kamalian, första författare på den här artikeln. En PhD-student från Iran som studerade 4234 artiklar, det vill säga allt som någonsin har skrivits om metaller och ALS i världslitteraturen. Och så hyvlade vi ner det där med olika kvalitetskriterier till att till slut analysera 29 stycken. Från vår egen studie till all lycka kom med då i det här gänget. Och från denna stora analys då så gick det att förstå att bly igen var förhöjt i ALS-CSF. Den diamond där är tydligt utanför. I sermoplasma var selen förhöjt och den passerar signifikansen. Så att bly och selen, återigen, när man gör det på det här sättet, när man tittar i litteraturen, verkar vara inblandade i ALS-pathogenes. Så ska vi lägga fokus på något som heter species. Vad är det då? Species betyder ju art eller sort av någonting. Och species i det här fallet handlar om bindningar till olika organiska molekyler av selen, om vi nu ska titta lite närmare på selen och dess roll i sammanhanget. Så här har vi då selenitionen, selenationen och så bindningar till olika organiska molekyler, metionin och glutathionsperoxidas, serumalbumin och framförallt selenoprotein P. Det här är italienska patienter, det är en studie av Vincetti som tidigt publicerade tillsammans med oss. Vi var först, jag bara markerar det. Vincetti kom strax efteråt och publicerade metalldata på ryggmärgsvätska ALS. Och då ska man veta som bakgrundsinformation att Italien är ett område i världen med höga koncentrationer av selen i berggrunden och i vatten. I kontrast till oss här uppe i Norge och Sverige som har låga koncentrationer. Och det här är selenitjonen. Den har 2 oparade elektroner och är reaktiv. Den här studien visade en direkt relation mellan koncentrationen av selenit och risken för att insjukna i ALS. Så selenit verkar man behöva ha uppmärksamhet på. Men det är en helt annan historia. Ja, förlåt, jag ska ta det här också. Selenit visar sig alltså ha högre koncentration bland ALS-fallen, ungefär dubbelt så mycket bland ALS-fallen. Och det intressanta med selenit är att den visar en specifik toxicitet för just framhållsceller. Och för att förstå sig på det så måste man gräva ganska djupt i litteraturen. Det finns en 5, 6, 7 artiklar som diskuterar den här specifika toxiciteten. Och bäst är den här då. Fütterungsbedingte Selenvergiftung beim Schwein. Bara det. Eller hur? Och den läser man då på tyska naturligtvis. Bilden visar ett tvärsnitt av ryggmärg hos gris. De här grisarna fick i sig selenit av misstag. Det var ett fel i fodret så att de fick en exponering för selenit av misstag. Och veterinären Aksel kom till grisarna och obducerade dem. Och här ser vi tvärsnitt av ryggmärg från gris. Och det här är framhornscellerna som är, som de skriver, fullständigt auflöst. Fullständigt upplöst. Det vita som går att se här. I viss kontrast då till baksträngarna. Det här är gracilis och cuneatus som för sensorisk information. Så tittar man bara på selenit i relation till skademönstret här så kan man ju förstå varför det handlar om en motorisk sjukdom men inte en sensorisk sjukdom. Så detta om selenit. Men titta här, selenat visar ingen korrelation alls. 0 och 0. Så att det finns ingen relation mellan koncentrationer av selenat och ALS-risk. Och sen de enskilda species här med olika bindning till organiska molekyler. Där kan man uppmärksamma det selen som är bundet till selenoprotein P där det är tvärtom. 0,8 mot 0,6 någonting sånt. Så att där verkar det ju möjligen skydda mot ALS då. Och vi som äter selentabletter till frukost kan alltså i lugn och ro fortsätta att göra det därför att Det är selenitjonen som är den otrevliga medan selen i övrigt verkar skydda. Då ska vi titta på det faktum att metaller verkar ackumuleras i framhornsceller. Och det här är 4 olika studier som är gjorda på råtta och mus, och det handlar den här gången om kvicksilver. Nu vet vi att selenit ackumuleras i framhornsceller. Det ser ut som att kvicksilver gör det också. Det här är tvärsnitt ryggmärg, det här är framåt, och här är ett utsnitt. De svarta kornen är kvicksilver efter respiratorisk exponering. Råttan fick andas in kvicksilverånga, och sen kan man hitta kvicksilver i framhornscellerna på det här viset. Det här är en annan separat studie som visar samma sak. Autometallografi, det här är en silver staining, så att det här är små silverkorn där det finns kvicksilver. Och se här, det är i cytoplasman men inte i kärnan som det här kvicksilvret verkar föreligga. Den här var kanske mest intressant. Det här är också råtta och inhalation. Det är lite högre koncentrationer här. Jag tror han hette Su, en kinesisk studie. Och här kan man se att de stora framhornscellerna vid den koncentrationen disintegrerar eller degenererar, samma sak där, samtidigt som de små och mellanstora är opåverkade. Det var också så i den här studien att framhornscellerna längst fram i framhornen var de som var mest afficerade, längre bak i framhornen mindre så. Och här är en 4:e studie där man försökte hitta kvicksilver i axonet och lyckades med det, så att kvicksilver ackumuleras i framhornsceller liksom selenit gör. Okej, sen när man kommit så här långt så vill man ju då utföra exponeringsstudier, eller hur? Nu skulle man vilja ha 103 medicinstudenter som man exponerar viljemässigt. De får andas in kvicksilverånga och selenit och bly och kadmium. Men det blir svårt, va? Det kommer inte att passera etisk kommitté. Ej heller i Norge, och däremot på häst så kan man göra det, eller kunde 2006. När den här studien är utförd av divers, en veterinär som matade 8 hästar med havre tillsatt med koppar och järn och jämförde med 51 hästar som hade fått vanligt havre. Och sen väntade man i nästan 2 år och tittade vad som hände med hästen. Och här ser man utmagring då. Den här patienten har väldigt tydlig muskelatrofi, håller huvudet lågt, dreglar så som ALS-patienter gör, autonom påverkan som ALS-patienter. Och den här hästen står ju fortfarande upp men kommer att lägga sig ner och dö. Medan däremot självklart ingen häst naturligt nog i kontrollgruppen blev sjuk. Och där tycker jag är nästan det kraftigaste argumentet för att metallexponering är inblandat i ALS-pathogenes häststudien. Ekvin ALS kallas detta, EQ-ALS. Vilka exponeringsvägar är aktuella? Ja, det finns respiratorisk exponering som vid vulkanisk aska. Man kan få det här i dricksvattnet och mat. Dermal exponering såsom tatueringar. Amalgamfyllningar i tänderna ger exponering för koppar, kvicksilver. Intramedullär, det vill säga det finns flera artiklar som visar koppling mellan ALS och benbrott. Och där har man spekulerat vad det kan bero på. Är det någonting med kalcium eller vad kan det bero på? Men obs då att benbrotten spikas med märgspikar som innehåller legeringar som innehåller järn, kobolt, mangan och arsenik. Så det är en exponeringsväg att beakta. Anekdotiskt höftproteser, patienter som får proteser och en tid senare, 1, 2, 3 år senare får ALS. Värt att studera. Intravenös exponering, axonal exponering, den så kallade retrograda axonala transporten. Det var Arvidsson, Karolinska på 70-talet som beskrev hur metaller vandrar baklänges genom axonet rätt upp i framhornscellen då, väldigt bestickande. Så gjorde man några radioaktiva experiment där och det lyckas inte reproducera där exakt. Men ungefär så här kan exponering se ut, och en självklar fråga till patienten du har framför dig då under anamnesen när du misstänker ALS. Vad har du jobbat med? Vad gör du på fritiden? Har du skrapat båtbottnar? Har du amalgam i tänderna? Har du varit med om benbrott? Och så vidare. Man måste alltså ta upp en noggrann exponeringsanamnes och vara medveten om alla de här exponeringsvägarna. Ett exempel på respiratorisk exponering, det här är en studie av Mount Etna på Sicilien som spyr ut då mangan, arsenik, vanadium och uran. Och downwind om vulkanen, det vill säga här, så är förekomsten av ALS högre än på andra sidan om vulkanen. Det blåser alltså därifrån till dit. The epidemiology of ALS in the Mount Etna region, a possible pathogenic role of volcanogenic metals. Så växlar vi spår lite grann och pratar om yrkesexponering. Jag kommer att beskriva flera yrken som alla är kopplade till en publikation, en eller flera som binder det yrket till ökad risk för ALS. Vi börjar med 4 tydliga sådana. Jägare. Det här är en studie från Los Angeles. Tänk The Deer Hunter, alltså de jagar kronhjort. Den är gjord av patologer och patologerna har studerat ungefär 100 fall, skickat ut ett frågeformulär. Det var en sak som föll ut, exposure to carcasses, det vill säga kroppar då. Och vad handlar det här om? Det var bara en enda faktor som föll ut och det var det där. Ja, när man skjuter ett rådjur eller en hjort så tar man ut kroppen och har kvar huden och den bestryker man med arsenik, så kallad arsenication. Jägarna exponerades för arsenik. Ett annat yrke, fåraherdar. Det ser ju stillsamt ut det här. Fåraherden sitter och tittar. Det finns en studie eller flera som kopplar yrket fåraherdar till ALS. Men så stillsamt är det då inte. Det här är fåraherdar. De ägnar sig åt så kallat sheep dip. Man doppar ner hela fåret i en blandning av rotenon, arsenik och svavel för att skydda pälsen mot fårlus, för att få bättre värde på pälsen. Sheep dip exponeras för metallen arsenik. Ett yrke med ökad risk för ALS är läderarbetare. Läderarbetare verkar ju stillsamt och fint. Nej, så är det inte. Det här är en läderarbetare. Det vill säga garveriprocessen innebär en exponering för krom och en exponering för arsenik vid färgningen. Gula, röda och gröna pigment är arsenikoxider. Att försöka få lite systematik i det här med yrkesexponering. Där har Gunnarsson gått igenom 110 publikationer och märker att exponering för kemikalier, insektsmedel och exponering för metaller är korrelerat till en ökad risk att insjukna i ALS. En annan metaanalys av Camel visade att insektsmedel, alltså pesticider, är kopplat till ALS-risk. Obs då att de flesta äldre insektsmedel är metallföreningar såsom blyarsenat, en kombination mellan bly och arsenik. Jag gjorde något liknande i min avhandling. Och de yrken som är kopplade, jag läser upp de här bara så får var och en fundera över vad det kan handla om. Ökad ALS-risk har alltså visat sig för jordbruksarbetare, för idrottsmän, Svenska Vasaloppet, flera ökad risk för ALS, piloter, men inte flygvärdinnor, arbetare vid elektricitetsverk, hårfrisörskor har högre risk för ALS, laboratorieassistenter, läderarbetare, verktygsmakare, krukmakare, arbetare inom medicinsk service, militärer, programmerare, gummiarbetare, tobaksplantagearbetare och svetsare. Finns det någon gemensam nämnare för alla dessa yrken? Och det här är resultatet av den metaanalys som gjordes. Den kombinerade relativa risken är 1,45 för att utveckla ALS när det gäller yrkesexponering för just metaller om man tittar på samlade data. Så metallexponeringen vid ALS verkar vara varierad, komplex, individuell och det verkar vara nödvändigt med en låggradig exponering under lång tid. Akut blyintoxikation som professor Zheng beskrev är ju någonting annat än en låggradig långsam exponering för bly. Två olika exponeringstidsfönster så att säga. Vi tar ett exempel till, verktygsmakare. Så vi letade upp en verktygsmakare i Stockholm med ALS. Och det var ett projektarbete för Sara Altonen, en medicinstudent. Och här ser vi fördelningen i ryggmärgsvätska av de olika metallerna. Till exempel aluminium fördelar sig i ryggmärgsvätska, blodplasma och helblod. På det där sättet i kontrast till bly som förekommer nästan bara i helblod förstås eftersom den binder sig till röda blodkroppar. Vi kliver förbi den där och går direkt på vad som behöver göras och det har vi där. Då har vi en hint om att det handlar om metaller. Vad är det som behöver ske här nu då? Ja, det här är lumbalpunktion under ultrarena förhållanden. Det där är jag inlindad i en hel massa skydds... Det handlar om att försöka göra en så ren och steril men framförallt kontaminationsfri provtagning av patienter med ALS. Och det här är på Huddinge sjukhus i Stockholm. Och vi försöker få så liten spridning på mätvärdena som möjligt genom att ha det så rent som möjligt. Och tar in kunskap från renrum och liknande. Det sitter HEPA-filter uppe i taket, dubbla HEPA-filter. Den värsta kontaminationskällan är patienten själv, så här har vi skydd. Jag jobbar med händer utan handskar. Vän av ordning skulle då säga ajabaja, du ska ha handskar. Men handskarna innehåller talk, talken innehåller metaller. Inte bra. Så har vi steriltvättat och syratvättat händerna för att inte kontaminera överhuvudtaget. Och här är sköterskan och det här går att göra nu. Det som behövs är mer provtagning. Kostar mycket pengar, behövs mer anslag. Så här, forskningssköterskan har också rent ögonskydd och ingen mascara och inget nagellack och så vidare för att få så ren provtagning som möjligt. Och vi planerar nu en fortsatt studie i Stockholm där vi tänker ta benbiopsier samtidigt och titta på ALS metallkoncentrationer i ben. Parallellt med en ny omgång, vi har fått nya anslag och vi kommer att använda oss av XRF för att separera metaller. Det är nödvändigt med ultraren teknik och det här är resultatet i ultrarena vials, syradiskade teflon vials. Och här ska komma en vial med ben. Slutligen, sista bilden. Jag ska säga lite mer om den här studien. Vi ber också om hjälp alltså med rekrytering av patienter till den här studien. Vi har fått rejält med nya anslag. Vi har en ny PhD-student som ska arbeta med det här de kommande 4 åren. Vi har det här renrummet ute i Huddinge och nya resurser, nya samarbeten. Så vi startar om lite grann här. Och behöver alltså flera fall som helst är nydiagnostiserade och inte står på ALS-läkemedel. Jag ska göra en sista utvikning, en parallell. Det här är Aksel. Det här är alltså en studie utförd i Finland på multipel skleros. Och det här handlar om sura sulfatjordar som är fördelade på det där viset i västra Finland. Och det här är utbredningen av multipel skleros. Vi intresserar oss för det tydliga överlappet här. Det här är den värsta hotspot för MS i hela världen. Med incidenser på 300-315 per 100 000, någonting sånt. Inom det område där De sura sulfatjordarna bildar svavelsyra som läcker ut metaller i omgivningen, vilket resulterar i fiskdöd och påstår vi också påverkar MS-förekomsten. Okej, övriga neurodegenerativa sjukdomar i relation till metaller, det får vi ta vid nästa möte med geokemisk sektion. Further reading, den som är intresserad, det här är en bok som heter Biometals in Neurodegenerative Diseases. Kapitel 10 handlar om ALS. Standardverket Handbook on the Toxicology of Metals, editor Gunnar Nordberg. Det är 2 rejäla volymer man kan läsa. Min avhandling på Karolinska Institutet, det är 90 sidor som diskuterar metaller i relation till ALS. Den är nu den mest lästa avhandlingen vid KI någonsin. Och tack.
So maybe I'll ask in English. I didn't quite catch the reasoning why you think these the increases in CSF metals you found was due to exposure and not some process of the disease itself. So when you showed that in intoxication, metals accumulated in the frontal horn cells that degenerate with ALS, you would expect them to be freed into the CSF, and couldn't that explain the increases you found? I didn't quite understand why you concluded that it was exposure.
I totally agree. There is no way that you can conclude that it is exposure at all. And the only way to disentangle this discussion is to use isotopic ratios or lanthanoid patterns and try to compare what is out in the environment and what is found inside of the cerebrospinal fluid. And very few such studies have been undertaken. And I think that is an avenue that has to be expanded. The Spanish study that I skipped, that's one effort. They tried to do that. They started with our findings in the cerebrospinal fluid, and then they went into the industry. And then they looked at the effusions from the industry in Spain and could correlate increases in ALS to increased exposure from industry. But to be 100% sure, you can't only rely on timed frames and things like that. You have to really analyze the isotopic pattern, both for lead and for other metals that have calculatable isotopes. And anyone who conducts that kind of study will forward this discussion a long way. So, yes, thank you.
Karin Persson, som skal prate om Alzheimers, nye medisinske muligheter. It's always a discussion.
And this will be a presentation that's quite different from the previous ones because it's no metals, not very technical research stuff, but more from a clinical view. So my background is that I'm a geriatrician and specialized in internal medicine and geriatric medicine, and I work part-time at the geriatric department at Oslo University Hospital and part-time in something called Aging and Health, which is a national center where we do a lot of research, but also educate the both specialist care, but also primary care on Alzheimer's disease and other things around dementia and other topics as well. So, but we don't have too much time now, so I'll just go through my slides. Little background about Alzheimer's disease in Norway, because some might not know how it's supposed to be diagnosed and taken care of here. So we have national guidelines developed by Helsedirektoratet in 2017, which are really good, and I recommend everyone to read them. And in Norway, most dementia diagnostics should be done in primary care, but younger patients and patients with more rare diseases should be diagnosed in specialist care. Then I also want to tell you that we have a national quality register for dementia in Norway, including patients from specialist care, which is called Norske. So we include patients from 47 centers across Norway and want more patients to be included into the register, both for research and for quality control. So what about the prevalence of dementia? That's usually mentioned in the media nowadays. So we have this animation which was made by Geir Selbekk, which I think opened at least my eyes some years ago. So based on the HUNT study from Nord-Trøndelag, we figured out how many people in Norway have dementia, and this was first what it looked like in 2020. And if you just kind of look at the figure, I think we don't need to look at the figures around here. Just look at the graph and what happens in the years to come. It's increasing and increasing because we get older. And the last one is what happens in 2100. It looks like this. So we have to think kind of like in the pandemic, how can we flatten the curve? And we have to think about ways to treat this disease or these diseases. But as you can see from the same examination in Nord-Trøndelag, here you can see the different etiologies. And we, of course, know that Alzheimer's disease is on top. We have vascular dementia, dementia with Lewy bodies, frontotemporal dementias, and some mixed variants and unspecified, which we want to be lower because we want every patient to get an exact diagnosis of the underlying cause. So what about Alzheimer's disease? As I said, 65% of all dementia is this disease. It's a slowly progressing disease where reduced memory, orientation, and word finding are usually early symptoms. Per Selnes mentioned that fatigue might be another early symptom, and there are others too. And what I also want to mention just before we talk about treatments is that there are many phenotypes of Alzheimer's disease. So while this is the usual symptoms that the disease starts with, you could have other forms that start maybe in the posterior part of the brain or the frontal part, and you can get language problems first or visuospatial problems. So there are different phenotypes. And maybe we should start thinking about metals more than what I will present today, or maybe the choroid plexus then. It gave me some new thoughts today. So that's the symptoms, and we know that prevalence increases with higher age, but it actually occurs already from 40 years old, which we see at the memory clinic at Oslo University Hospital, where we focus on the younger patients. So that we know the genetic risk, which you can inherit, and the pathology, at least what we usually talk about, is that we know that the patients have amyloid plaques, we have misphosphorylated tau proteins, and we have inflammation. And that's what Alzheimer found in the beginning of 1905 or something like that, when he looked at the brain of a patient that he had. So we don't know why this happens to the actual patient. Maybe we'll know more in the future, but that's what we find in the brain. And the amyloid hypothesis, which is the one that I think most people believe in right now, is like this. So this is a graph just showing time and disease stage on the x-axis and the biomarker, how they appear along this route from normal to abnormal. And so it starts, many think, with the amyloid plaques building up in the brain. And this can be found using, you can do CSF, or you could do amyloid PET, and you can find that this has happened. And then you get the neurofibrillary tangles in other parts of the brain, which you can find both using tau PET, or you could also do FDG PET to see that the neurons are starting to dysfunction, so you get a lower uptake. Eventually, you get atrophy of certain parts of the brain. And first, when you come to this stage, you actually get symptoms from the disease, and you get problems with your cognitive tests, and you need help in the daily living. So you actually have a dementia. So it's a progressing disease where amyloid and tau and inflammation are important. So what I wanted to talk about today, or what I thought was one interesting thing, is, of course, the new medications that we read about in the newspapers now, which is mostly targeting amyloid. That's what we read about right now. So it started in 2016 with, or before that also, but the first agent was aducanumab, so monoclonal antibodies that are injected into the blood, and that actually removes the amyloid depositions in the brain. So that was kind of the first breakthrough, where they found that this aducanumab could actually remove the plaques. So from baseline and for 1 year with placebo, nothing happens. And then with increasing dosages, you can see that the red stuff, which is the amyloid depositions, are totally vacuumed from the brain. But unfortunately, this first medication didn't show any improvement of the patient. So why was that? The pathology was removed, but the patient didn't get better. And so then new medications have come to the market, and we know that these are FDA approved, so in use in the US. Maybe you know more about the practical use nowadays, but in some other parts of the world also these are in use. And very recently, in November 24, Europe decided to approve the first one, the lecanemab, after some discussions for and against. And right now it's discussion in Norway if we should or if they would reimburse use or not. So it's a very, everyone is waiting for more knowledge on, or to know what they decide on. And we get phones every day from patients that wonder when they can get this treatment. And so what is the treatment? So these are just graphs of these two agents, the lecanemab and donanemab, and both these agents have shown that they effectively remove the amyloid. Placebo, nothing happens. Lecanemab, it's doing what it should. And then the new thing with these two agents is that they actually also could show that the patients got better. But they did not get better, actually. They just didn't get as worse as those getting placebo. So you can see that the blue ones are placebo, and the two agents are really quite similar in the effects. So what we see is that patients all get worse, but those on the active MAB doesn't get as much worse. So in 18 months, while the studies were going on, you could say that they were kind of saving 6 months of getting worse. Does that make sense? So a person, you kind of slowed the progression. You would, of course, the best thing would be that they didn't get any worse at all. So we have to think, why is that happening? And that could be many reasons. It could be that there are more pathology than just amyloid driving the neurodegeneration. It could be that we come in too late with the medications. Maybe the process has gone too far and it's difficult, or you cannot kind of stop everything. Or it might be that the agents aren't perfectly stopping the process going on. So we don't know what will happen if you give the medications for more than 18 months, and that will be interesting. but it seems they do remove amyloid. Patients get somewhat less worse, but we also find that there are many subgroups that seem to have effect of the medication, and some groups don't. And it looks like, for instance, if you have ApoE epsilon 4 allele, which is usually, if you're homozygous for that allele, you are very at high risk of getting Alzheimer's disease. And they are actually the ones that do not have effect of the medication, it seems. And also you could see that females are not as, doesn't have as much effect as males. The young patients, where we would expect maybe the best effect because they might be maybe at an earlier stage, they are not, do not have an effect due to these studies. And also, some ethnical differences could be found. So there will be a very small group of patients that are actually the ones that could seem to benefit from the medication. And there are side effects and obstacles with the medications. That's what the governmental discussions are about now, is the cost and the effect. Does this match? Is it worth it? And also with side effects. Because to get this medication, you would need to know that you have amyloid in your brain. So you would need a CSF analysis or an amyloid PET. But we also get new plasma biomarkers soon. So that's an important news that we could talk about as well. The medication is intravenous. It costs a lot. And we have the problematic side effect with amyloid-related imaging abnormalities, which we don't... Actually, about maybe 20% get these changes with edema or small bleedings, and most of them are not... don't seem to be very serious, but still we don't know the long effects of that yet. And you would need frequent MRIs to go through this to use this medication, and also the medication would need to be given for some time, and then they've seen that you could maybe have a pause and then keep on with a new treatment period, because the amyloid will build up again if you stop with the medication. So who's it for? It's for something we call mild cognitive impairment, which I haven't talked about, but that means kind of an early stage of Alzheimer's disease or the mild dementia. It cannot be too late. You could not be homozygous for the ApoE4 allele, which means that you have to go through genetic testing, and that might be an ethical problem for families that they also have to think about. You should not be on anticoagulants, because then you are more disposed to these area side effects, and no cerebrovascular comorbidity. And there's a recent paper that looked at European situations and what introduction of these medications would mean, and it's important to know that the impact of the approval might have very large consequences to the healthcare systems, and we have to prepare on how to discuss this with patients, and also we have to think that the research should not stop because we get the medication like this, because we still don't know everything about Alzheimer's disease. So I'm thinking a little bit like this little person: who is this medication for? And who should I be the one taking the genetic test? Should I do a plasma biomarker test if I know that I might get a disease in 10 years? Many questions to think about. And then for the last slides, I just want to mention that these monoclonal antibodies is definitely not all medication trials going on. This is an overview that comes out every year that shows all the phase 1, 2, and 3 medications being tested. And as you can see, there are many different agents. You usually divide them into disease modifiers, cognitive enhancers, and neuropsychiatric symptom medications. And there are different, not only monoclonal antibodies, but many other forms of action points, which is more of the stuff that you have been talking about than what I know a lot about. And also how to flatten the curve. It's important to know about prevention. So that's has a lot of focus in media also. How can we prevent more people from getting dementia? And that's from Lancet 24, where maybe some of the new risk factors was, for instance, hearing loss. Everyone in their mid or late life should use a hearing aid. And of course, the regular cardiovascular diseases should be prevented and keep active. So that's a little bit from the clinical side, I guess.
Questions?
About medications for ALS. Are there any?
So then I have to give the word to the neurologist.
The neurologist, one or another?
One or the other, yeah.
I guess.
Well, it's a similar situation with the pharmaceuticals that you can give for ALS. They have limited effect like this, and you can, as you put it, buy a few months or something like that. And some patients want that and accept that, and some of they refuse medication. So that's the situation.
I guess you discussed memantine in Karolinska, at Karolinska. Yeah. Marginal effect.
Well, we know right now it's the an antibody is frequently used. How about this the traditional way treated Alzheimer's disease use the like the M receptor in acetylcholine kind of a drug.
You mean
That's
symptomatic
yeah.
medications? Cholinesterase I don't know the English word, but acetylcholine esterase inhibitors, maybe. Yeah, so that's what we use a lot. Yeah, but that's more like symptomatic treatment. I guess I recently read some new papers that there might actually be a little disease-modifying effect with that as well, just because you kind of activate the brain when you use the symptomatic medications. So I know from Sweden, I think they came. So I think we in Norway we have become more liberal with giving out those medications.
So are there any combination therapy, use antibody to take the beta amyloids out and in the meantime treat for the symptoms?
Yeah, that's what happens, I guess, in the US when they've started with the antibodies, that you can do both. And I think probably in the future we will have to treat many pathologies at once, because it obviously is not only amyloid, or maybe you have to start very, very early. Yeah.
Well, I have the other question. So does the antibody treatment the male versus the female, is there any kind of a sexual difference, or the treatment is all the same, the outcomes are the same? Or maybe it's better on the females.
So it looks like it's better for males, right? Yeah. I don't know why it's that way. There's also another sex difference, because more women get Alzheimer's disease than males. So is there another pathology process going on? Do you know? No, I don't have an answer to it.
Thank you, all of you in the audience, and now it is closed.